So for identifying these translational products, that is your proteins from these cloned genes, there are two techniques. One is called as your HRT. HRT का मतलब क्या होता है? Hybrid release translation, hybrid release translation. Whereas you have HART, where it is hybrid arrest translation. So these two techniques are very related to each other. So there are some minor changes which we will be discussing. Okay. So they will be used to identify whether your that cloned protein is being synthesized by your whatever your recombinant cells or not. So both will depend upon the ability of the purified mRNA to direct synthesis of proteins in cell-free translation system. This is very, very important. What is your cell-free translation system? That means here we are actually not going to use any kind of cells for the translation. Okay, here the translation will be taking place in vitro. Matlab, whatever the proteins and whatever the requirements of are there for the translation, so all those requirements you will be getting in vitro. Okay, you will be getting in a plate or in a tube, and that those whatever components are there, that will be bringing about the translation. It is not going to be in vivo in nature. So you have cell extracts. Okay, there would be your cell extracts which will have all the requirements for your translation. Okay, so these cell extracts, what they will do is okay, that will be prepared usually from your germinating wheat seeds. Okay, they will be getting whatever your proteins or whatever the requirements for translation. So they are mostly your germinating wheat seeds or from your rabbit reticulocytes, because these proteins whatever are there from these sources, they are very, very active in your protein synthesis. So to have this for the working of your this particular method, having proper active proteins or having proper active protein synthesis is very, very required. So that will have these cell extracts will contain your ribosomes, your tRNA, and whatever the other molecules that are required for your protein synthesis. So what we will be doing is the cells, the recombinant cells from which we want to check whether it is capable of producing our proteins or not. The mRNA would be extracted from those cells, from your recombinant cells, and those mRNAs are going to be added in this whatever your cell-free translation system is there. So cell-free translation system, which includes the cell extracts from your germinating wheat seeds or your rabbit reticulocyte cells. So whatever your cell extracts are there, your mRNA from this recombinant cells are going to be placed in these whatever your system is there, along with 20 amino acids, that which are responsible or which are found in the proteins. We all know that there are many amino acids that are there, but amongst that there are only 20 which are found in the proteins. But out of this 20, one would be labeled. Okay, one would be labeled by a higher isotope. For example, methionine. If it is methionine, we can label it with 35S. Okay, so this is for the detection. For the detection purpose, we will be required. So methionine is preferred because you know that most of your first codon which is there in your proteins, it is your methionine. So most of your proteins are going to get your methionine. So that is the reason why we can use your 35S labeled methionine for this particular purpose. So the mRNA molecules which are going to be translated, okay, so what will happen in this system, from this system, the amino acids would be incorporated in the proteins which are synthesized, right? So in this incorporation, that one labeled amino acid will also be included, and that is the reason why what will happen is that your entire protein will get radio labeled. Okay, radio labeling would be there. And so we can separate out these proteins using your gel electrophoresis method, like your page or native page also, and then afterwards we can visualize it using your autoradiography. So each band will represent a particular protein which is coded by one of the mRNA molecule which is present in the sample, right? Your cell, whatever your recombinant cell is there, so whatever the proteins, okay, the whole set of your proteins which are synthesized at that particular time in that recombinant cell, so the mRNA corresponding to all these proteins are going to be present, okay? So that is going to be present in the sample, and those mRNAs would be translated in vitro to your all the proteins in vitro, okay, using your this cell-free translation system. It is going to be in vitro. So let us see. So what you can see over here, this is specifying your entire process of your cell-free translation. Kindly note, students, here we are talking about, or here I am first explaining you what is cell-free translation. I'm still not started with HRT or HART. I'm first explaining to you about your cell-free translation. Okay. So you have your purified mRNA. From where this purified mRNA is coming? It is coming from your recombinant cells. Okay? That purified mRNA you are putting into the cell-free translation system. So what does this cell-free translation system contain? It will contain your all the ribosomes, your tRNA, all the proteins, all the molecules which are required for your translation, right? Along with your 20 amino acids. And one thing has to be taken care that so in that 20 amino acids, one amino acid is going to be radio labeled. So here we are showing that it is your 35S methionine. So it is a higher isotope of your sulfur. So that is that higher isotope containing sulfur is going to be present in methionine, right? So when your mRNA and this entire system comes in contact with each other and they are mixed with each other, the protein synthesis will start. So here you will see that your this higher isotope labeled methionine gets incorporated into your proteins, okay, and they will get labeled. So then once this all your proteins, so every mRNA is going to give rise to your labeled proteins. So agar tumhare isme there are say 15 different types of mRNA, then you are going to get 15 different bands. Every band corresponding to one protein coming from each of your mRNAs, right? So you will get, you will do the gel electrophoresis. So you are going to get your bands, so which can be run using your, along with your molecular markers, so that you can find out the, whatever your, you can find out what is the size of your cells. Sorry, what is the size of your molecular size of your proteins? That can be definitely understood. Okay. So then, because they are radio labeled, so what is going to happen is your using your autoradiography, you can get those specific bands, okay, which can be stored for a longer time for the next whatever your studies that are there. Okay, just a second. So that is how your cell-free translation system is going to work. Any doubt regarding your cell-free translation systems? Have you understood, students? Any doubt? Okay. So let us move ahead. Now we will understand how using this cell-free translation system, your HRT and HART will work. Okay. So most of the times, one thing, just excuse me. I'm so sorry. So both your HRT and HART, they will work best when the gene, okay, when we are inserting it as your cDNA. Why as a cDNA? Because you know that most of the times we are studying, or these recombinant cells or your host cells are going to be your prokaryotic cells, like your bacteria. Okay, so they don't contain this splicing mechanism. Yes, because they don't have any kind of your introns in their system. So if we want to have, any kind of your eukaryotic protein to be, cloned or being expressed in these cells, so it is very important that your what you are cloning should not be the exactly the gene, it should be the cDNA, because cDNA will not have the introns in it. So proteins, if you want the proteins to be synthesized and to get the actual, actual form of your protein, so definitely intron should be spliced off. So that is the reason why it should be your cDNA should be there. So for HRT, the cDNA is denatured, immobilized on a nitrocellulose or your nylon membrane, and incubated with the mRNA sample. So what is going to happen in that case? So cDNA, which is your what you can say, which is your complementary to your mRNA of your interest or the protein. So you are selecting whether your protein is synthesized or not. So for that protein, you will have an mRNA. Agreed or not? Yes. Now you have an mRNA. So mRNA ka complementary, you have cDNA. Complementary would be your mRNA. Agreed or not? Yes. So what you will be doing is, so you would be immobilizing on your nitrocellulose membrane, you would be having your cDNA, okay, a single stranded cDNA, it is denatured single stranded DNA, and whatever your mRNAs you are extracting. So here we are talking about only HRT, okay, so this is the process of HRT. You have to understand what is the difference between HRT and HART. So what is going to happen is this, whatever your cDNA is there, so that cDNA is going to be incubated with the mRNA sample. mRNA sample coming from your recombinant cells. So this specific mRNA counterpart of the cDNA hybridizes. Yes, matlab specific mRNA matlab kya? That mRNA of our interest, which we are finding, which we are trying to see whether the cell is expressing or not, right? So we are going to use the cDNA, which is complementary to that specific mRNA. Agreed? So that specific mRNA will get bound to that cDNA. cDNA is already attached to the membrane. So you can see that membrane hai, membrane ke upar you have your cDNA. So that cDNA now gets bound to your mRNA. So you can see over here, you have your membrane. To that membrane, what is coming and what is there? It is your specific cDNA, the cDNA to which we are trying to find out the complementary mRNA to. So when the mRNA mixture is applied, only the mRNA which is complementary to this cDNA that will go and bind. Yes. Okay, then you give a wash. So whatever the unbound ones are there, unbound mRNAs are there, they would be washed off. Okay, so what you will remain back is only the mRNA hybridized with your cDNA. Okay, so that remains attached. So after discarding the unbound molecules, the hybridized mRNA is recovered. So now this mRNA, which is hybridized with your cDNA, only that is removed. Only that is removed. So only that particular mRNA is going to be put in that cell-free extract, जो हमने यहाँ पे देखा. So in this cell-free extract, okay, along with the cell-free extract. So here what we are talking about the mRNA, pure mRNA, just understand here you are having only that one mRNA which is hybridizing with your cDNA that you have added for the selection. Yes, so only that specific cDNA, say jo bhi whatever your hybridized mRNA is there, so only those molecules of your mRNA is going to be added into your cell-free translation system, and then it will undergo electrophoresis, and then you will be getting your autoradiography. So if you get a band in your system at the end, if you get a band, what does that mean? That yes, the cell, the recombinant cell was producing the mRNA, which is complementary to your specific whatever your cDNA that you have added for selection, and the protein is synthesis. मतलब यहाँ पे एक band आना मतलब it tells you that yes, the cell, the whatever your recombinant cell is there, it is actually synthesizing the protein which is encoded by that particular cDNA. Yes, students, is it HRT is clear to all of you? A quick yes or no. If you want me to repeat, just write a repeat in the chat box quickly. Anybody who wants me to repeat? Okay. Okay, Bhavika and Vishnupriya, I'll repeat. No problem. So, what we are saying over here, यहाँ पे we are trying to find out whether the cell, your recombinant cell, is producing your protein of interest or not. Okay, so how do we do it? So we will have, so protein of interest, you know which is your protein of interest. So definitely you know which is the cDNA which would be actually producing that your protein of interest. Okay. So what you are going to do is you will be taking that cDNA, that specific cDNA, which is complementary to your protein of interest or which is complementary to your mRNA of interest. ठीक है? So जो भी तुम्हारा nylon membrane रहेगा, so on that your cDNA is going to be bound. It will be bound. ठीक है? Now what you will do is you will extract out the entire mRNA from your recombinant cells. सारे के सारे mRNA are going to be extracted out. So that is your mRNA mixture. Okay, now that mRNA mixture you will allow this nylon membrane to hybridize with. So what will happen if your mRNA of interest is there in this mixture? What will happen? That specific mRNA will go and bind to your cDNA. If it is not there, what will happen? The mRNA, there would be no mRNA that is binding to this nylon membrane. Yes or no? Yes. So after this hybridization has taken place, you will give a wash. So whatever your unbound ones are there, they are removed. Okay, iske baad you will give a wash so that the unbound ones are removed. Once the unbound ones are removed, now you will unhybridize. So what you will do is remove this hybridized mRNA. Okay, इनका जो hybridization है, you are going to remove it. Okay, so remove that. So only these mRNAs which are being bound to your cDNA, that is removed, and those mRNAs are added to your cell-free extract. Okay, whatever we had seen in this particular method, the same. It is added to your cell-free extract along with the 20 amino acid, along with this labeled 35S labeled methionine. So what will happen? Only that particular mRNA would be now producing your labeled proteins, and so that will undergo your gel electrophoresis and autoradiography. So if there had been any mRNA which has been hybridized to your cDNA, then that will produce your protein and give you a single band. So getting a single band suggests that yes, the recombinant cell was able to produce your mRNA of interest. If you don't get any band on this particular electrophoresis or your autoradiogram, what does that suggest? That your recombinant cell was not able to produce the protein of interest or your mRNA of interest. So that was the reason why there was no mRNA that got hybridized to your cDNA, and hence the cell-free extract was not able to synthesize any proteins, and hence you are not getting any of your band. Now is it clear, students? Everyone? Vishnupriya, Bhavika, Trisha, Sabha. Hivani, is it clear? Okay. So now let us move on to the next method, that is hybrid arrest method. Okay, so it is slightly different in that the denatured cDNA is directly added to your mRNA sample. There is no nylon membrane used in this particular sample. Okay, so you have, say for example, you have your tubes. Okay, in that tube you will be adding whatever your cDNA is there, which is specific to your mRNA of interest. जैसे हमने the one which we have used for your HRT, the same cDNA you are going to use with this, and then you add your mRNA sample which is extracted from your recombinant cell. Okay, so ये देखो, that is how you have your mRNA preparation from your recombinant cell, which is added to that specific cDNA. So what will happen? Your cDNA will get hybridized to the mRNA counterpart, whatever your specific is there. Okay, now, so out of this entire lot of your mRNA, only this one, only your mRNA of interest is hybridized, right? So it is double stranded in nature. It is double stranded in nature, whereas the other mRNAs are single stranded in nature. Now tell me that in translation, what is required? A single stranded mRNA or a double stranded one? Can double stranded mRNA undergo direct translation? Understood the difference? Okay, so agar in this mixture, if you have your hybridized one and unhybridized one, now tell me which one will undergo translation, hybridized or unhybridized? Which mRNAs will undergo translation? Unhybridized. Very good. Yes, only the unhybridized ones. Okay, यहाँ पे जो ये single stranded mRNAs दिख रहा है, they will undergo translation and produce, yes, they will undergo translation and produce your proteins. Whereas this one, which is your hybridized, this is your protein of interest or your mRNA of interest, which has got hybridized with that complementary cDNA. So what will happen? What will happen? That will not get translated. Okay? So if say whatever your proteins that you are getting from this particular thing, you will run on one well of your gel. This is one part. Now on the other well, what you will do is the same thing you will do, but you will not add your cDNA. Yahan pe you will not add the cDNA. So what will happen? All the mRNAs which are there, that would be added to your cell-free extract or your cell-free translation system, and all the proteins are going to be synthesized, right? So one well will carry only the HART, arrest, HART, hybrid arrest translation, right? So hybrid arrest release translation. So arrest kar that means you are not allowing the actual recombinant protein or your clone protein, clone mRNA to get translated. You are arresting the system. Okay? So whatever your HART product is there, that will be allowed to run on this first well. Whereas total translation products will be allowed to, total translation means what? Where the entire mRNA preparation is there, that will be allowed to run on your or that will be added to your directly your cell-free translation system. So all the proteins are going to be there. So now compare both the banding system. So here you can see what is going to happen. So here you will be able to see that, I'm so sorry, just a second. So this particular band is missing from here. है ना, बाकी सारे bands है, all the other bands are there, but this particular band which you are able to see in this total translation product, that particular band is missing from here. Yes. So what does that mean? That this is the protein. This is the protein of your interest. Yes, which is arrested in this particular whatever your so sorry. So that is getting arrested. Okay. So from your HART thing. Okay. From your whatever your procedure of your HART is there, it is getting arrested from that. Understood, students? Should anyone wants me to repeat? Kindly let me know. Everyone has understood the difference between the two? In one method, in HRT, we are directly finding out the mRNA of your interest by using the hybridization. Okay, cDNA or hybridized mRNA को ही हम select करते हैं, and then we are trying to put that only into your cell-free extract, right? Whereas in case of your in case of your HART, here we hybridize it with your cDNA. So because of the hybridization, it will not undergo the protein synthesis, and that is why that specific band is going to be missing. That specific band is going to be missing. Yes, students, is it clear? Everyone, anyone wants me to repeat?